Yeah, good evening to all of you who have joined the live session. Today we will be discussing about um, the topics that we covered in the last 2 weeks, and we'll take up some of the questions that are already there in uh, the Google form that um, we have collected. And you also have the option of posting the queries using the chat box, the YouTube link that has been given. So we will try to address as many questions that are there. But my request would be that we would only consider those questions that are technical, that is about the syllabus, about the topic that we have discussed. We perhaps will not get into questions that are related to the exams or about your assignment that you have missed and so on, because that is not handled by us. It is handled by the team, that the NPTEL team, both from IIT-Kanpur and IIT Madras. So you may want to send mail directly on the forum. It will be attended to, but that is not something that we would be addressing. With that little background, I would start answering some of the questions that are there already on the portal, the Google form that has been collected. There are quite a few questions. Maybe the first question, I'll take it up. The first question is how functional genomics helps medical industry. It is, you need to understand functional genomics is a tool, and you can use a tool for a variety of applications, just like knife. You can use it in your kitchen, or you can use it for sharpening your pencil, or for surgery and so on. That's what a tool is meant to be. And you must have seen during this COVID pandemic, right from the diagnosis, whether you are COVID positive or not, the RT-PCR, to typing the different variants of the COVID virus, saying this is a Delta virus, or this is more virulent virus, this is a South African variant, and so on. All these information that we have come from functional genomics approach. Basically, they do a RT-PCR and then do a sequencing to understand whether there is a sequence variation, and if there is a variation, then is it similar to the one that has been reported elsewhere, or is it different? If it is different, it's a new variant. And you can compare the variant with the symptom that a given patient was showing to map and say whether this is far more aggressive form of the virus or less virulent, and so on. So I'm just giving you one example as to how this can be used there. That's what you mean by medical industry. If you're talking about this is Ganesh Raj. If you're talking about healthcare or medicine and healthcare sector, that is what the application is. But if you're talking about pharmaceutical industry, it's a very different kind of applications. You must have seen that we have discussed about how we can use high throughput method for drug screening and so on. So that's a different. We are talking about therapeutics. It's a different applications. So as I said, again, this is a tool. It can be used for a variety of application depending on the need and your ideas. There's a question from Palash Kumar Sanyal about the textbook that we may recommend for this course, functional genomics. Since the topics that are covered are contemporary in nature, many of the approaches that is currently being used, we may not find one textbook that covers all the topics that we discussed. That is, if you are interested in finding more or you want to read more about the topics that is covered in this course. But what you have done is we have given, all the links and other, where this is publicly available, that is being given as part of the lectures. That is good enough for most of the reading. However, you wish to read more on that, and most of the molecular biology books that you have, and the books that are having a topic of genomics can be used. Again, that would not cover everything that we have covered in this course. So I would say, if you want to have a book that covers every topic that we would discuss in this course, you may not get a book like that. But any book that talks about genomics would cover part of it, right from, for example, all the cloning strategies, sequencing strategies, how do you create libraries and knockout lines and so on. But more recent application that we discussed perhaps is not available. So you have to have multiple books. There is a question about that about the course being starting on 30th August, but and so on, but about assignments and all. So I would refrain from answering these questions. This is something that you may wish to write to the, use the course portal and put your questions there, or doubts or clarification, and the office would be able to help you out. I'm not involved in day-to-day operations of the courses, so that I would not be able to help you in this. That's another question is, what are the pros and cons of PCR and cell-based cloning? There are many. It depends on what you wish to do. And PCR can be used only, if you know the sequence of the target DNA that you wish to amplify. Then you have to have a specific primer, then you will be able to make multiple copies of it in quick time with very little investment. That's the application of PCR. But if you have created a library for which you have the DNA molecule, but you don't know the sequence, then you have to go for the cell-based cloning. I believe you refer to the transformation, the traditional way of cloning, that is transforming the E. coli with the ligated vectors and then having separate colonies and selecting each of the colonies and sequencing and so on. So that's the approach. So that's, these two are different. In one, we need to know the sequence for you to amplify using PCR. The other one, you did not. If it's a heterogeneous mixture of DNA and you wish to use the primer from the plasmid, the backbone, then it is not of any use, because you're going to amplify every plasmid. That will be a mixture of all the DNA that will not give you any information. So that's the difference. The other question is, what's the difference between siRNA and shRNA? The difference is simple. If you are using a synthetic RNA strand, which is double-stranded, which you have produced using a machine like oligonucleotides, and you're using that to transfect the cells to silence the gene of interest, then it is siRNA. But if you're using a plasmid-based vector, which has the sequence, which when gets transcribed and makes a short hairpin-like structure, which is having a sequence identical to the target sequence, and you use the endogenous cell machinery to make that kind of hairpin structures within the cell by transfecting the cells with the plasmid having a promoter, then that is shRNA. Now, there are pros and cons. For shRNA to be made, then you have all these plasmids to be generated. So you have to clone and so on. That's going to be time consuming. But if you're using RNA with a synthetic RNAs, which can be produced in bulk amount, then the time required for you to make them is less. So that's the difference. The other difference is some people say that the plasmid-based RNAs and ShRNAs are far more um, robust in terms of silencing the gene of interest, whereas SiRNA probably is not as good as the shRNA. So these are some of the differences. Again, it depends on what you wish to do and what are the avenues that you can use to create these things. That's the answer in short. There's another question about the history part of the era of molecular biology. It's confusing, and that we explained in a nutshell. It's a history, so explaining them in a nutshell is not going to be easy. But what I wish to reiterate here is that that how over the time from 90s to 96 and later, how like up to maybe 2003, 4 when the genome was sequenced, how the genomic approaches evolved and how that really helped us to develop the new topic called genomics, right? So it all started with the Human Genome Project in 90. That basically started with identifying the genes that are defective in a genetic disorder in families and find the gene, what you call as a positional cloning. That is how the whole thing started. And then people understood that identifying all the genes in the genome might help us to understand the genetic disorders. So they went ahead and launched the big program called Human Genome Project. That led to the formation of several consortia and then making genomic libraries. So that is how the bacterial artificial chromosome, yeast artificial chromosome, or cosmid libraries, they have come in. They develop these vectors. They're able to put large segment of human genomic DNA into them, and then making them into smaller DNA and of course, when you have a larger DNA, you are able to map it to a given chromosome. Therefore, you know which chromosomal DNA you are dealing with. Then there are labs that are assigned with different chromosomes. For example, Lab X looked at chromosome 11 sequencing, and another lab looked at chromosome 10, and so on. So they have sort of distributed the work, and then each of the lab made what is called as chromosome walking, identifying the shared regions of the DNA and then sequencing, and then they have created the kind of a library and sequencing approaches. And then, of course, there are many other things that happened as part of it. Once the human genome started, human genome sequencing project started, they also began working on other microbial and other genomes sequencing approaches. So therefore, that's the way the whole thing started. And of course, recently they have went ahead and mouse as well as the other microbial sequences and so on. So, but in this process, what has happened is that they came up with approaches and also instruments that can take up this kind of high throughput sequencing approaches, right? So that's what it is from 1990 to 2003. This whole um, um, the whole discussion in the slide were about the genomic era. So that is what it is. But I think if you look at the slides there, I had given a link that is genomicsforeveryone.org. So you can go and search this. This is http:// or you can give this as a search command, genomicsforeveryone.org, and then it sort of gives you the entire the genomic era, the 20 years, what has happened, what are the milestones. You'll be able to read and understand better. So that's what in nutshell that I can tell. There's another question called why we are using rat as a model for experiments on human disease instead of monkeys. And monkeys are more closely related to the humans. The most common model for genomic application is not rat, it is mouse. There's a difference between rat and mouse. Mouse, people use it because there can be a lot of genetics can be easily done, and we know, of course, the genome sequencing, and then they have a shorter gestation period. Once they are born, in about 50, 60 days, they become adult, and then you can set up cross, because genetics, we need to look at F1, F2, F3, you have to set up breeding. So that is easier if you are using mouse because of short lifespan and reproductive cycle. But monkeys live like 40 years, 50 years. If you really want to look at genetic, for example, disorders as a model, or what are the genes function and so on, you have to manipulate the genome, right? And if you manipulate the genome, you have to look at the next generation, because you create most often these kind of mutations in the germ cells. And then if the germ cells are allowed to fertilize and form a young one, then the young one develops, and then you look at what happens to that animal. So that can be studied only if you are looking at the second, third, fourth generation, so on. So monkeys are not ideal in that, because they take 20 years to become adult and have next generation. So even in a lifetime of a scientist, assuming someone become an independent scientist when he or she is about 30 years, probably at most by the time you retire, you'll be able to see one generation. So that is not something that you desire. So that's where mouse is a good model system. But of course, monkeys are used for cognitive functions. When you talk about how the human brain functions, if you want to understand some of it, many of the cognitive abilities kind of experiments are tested using monkeys. There's a question, since the lectures talked about forward and reverse genetics, my question is, in case we know that certain metabolite, not protein, is being produced by an organism, then how we can identify the gene responsible for the production of that metabolite. When you talk about metabolite, is one byproduct of metabolism pathway. And if you talk about any such pathway, there are a good number of enzymes involved. It's not one, it is maybe 5, 6, 7 enzymes. So what we normally looked at is that if a given enzyme is defective, then that particular byproduct is not converted any further. Therefore, the level of that byproduct increases. So this is something that it is known already. There are certain drugs when we take, for example, the enzymes that are there in your body metabolize these drugs, and this metabolite is the one that is functional as the drug. It is not exactly the same compound that you take. Some people have some issues, in the way the enzyme is active in your body, and therefore, because of the defect in that particular enzyme, this metabolite may not be produced, or it is not converted further. It can become toxic. It is known there are certain conditions that is known, and it is known what enzyme involved. So it depends on which metabolite you are talking about, and if it is known what is the synthetic pathway for that metabolite. It is there in the literature. It's not that difficult. One can look at it. Yeah. In fact, some of these classic urine tests are done by that kind of a mechanism. I don't know whether you are aware of what phenylketonuria, like it's also a defect in enzyme, right? So that is known. Why Indian funding agency has not taken initiative to establish an indigenous repository comparable to Gene Bank or PDB? It's a good question. It all depends on how much money you have to invest. If you are talking about a gene bank, then it's not about simply having a server to store your sequence data, but your wherewithal and your infrastructure to generate these sequence. So we are not pioneers in making these machines that can make large amount of sequence data. Therefore, the other countries started, like the US and then Europe, and then Japan. So these are three major databases that you have. And they have mandated that anything that you generate here, you have to submit in one of the databases. Now it's all redundant in the sense that it's all freely shared between all the major databases. There's nothing really prevents us from accessing any of the data. That's simply the reason. It is not like, India, there's no point in investing now to have a server to have the data now, because this is anyway available for most of the genome sequences. But India was part of this RISE genome project. So the RISE genome, for example, is important for the South and the East Asian countries. So it's from Middle East to Japan. These are the countries that have rice as a staple food. So there was a consortium that developed to sequence different varieties of rice, plants, and they have tied up and they created their own database. Some of them are housed in India. So, but classic human and others, that is not there. Yeah, that's the difference. And yeah, so that's the question that we have so far in the Google form. Maybe now I'll look at questions that are from the YouTube itself. Right. So Vijay Kumar Singh from BIS, that I have already responded to, that why there are no repository comparable to GeneBank and PDB, that we have already done. We are looking at other question. A general doubt: when do we classify a microorganism as different strain? How different does one organism have to be from another to be a different strain? Yeah, there are. This is not only a functional genomics question, because the different species and their classification is not something new. This is done even before the molecular biology came. So those days, this field was called as taxonomy, right? So there are beautiful classification that is done. You have phylum, class, genus, species, and so on. And it is purely based on the morphology, the development, and many other things. So that is looked at. And there are clear criteria people have used to classify and say a given plant is a strain, that is a milder variant from a species, equivalent to, for example, races in Homo sapiens, for example, Africans, Asians, Chinese, and so on. And and it is not strictly you do a sequencing and compare and say. It is more at the morphological features as well. For example, the best way that you can define that two different plant or two different microorganisms are distinct from each other is that they, if there is any sexual reproduction, they cannot reproduce between them. So they have a very different kind of cell structure. They have very different kind of their physiology, their survival mechanism, susceptibility to any of the antibiotic that you do, and so on. So that's the way people do. It's not necessarily based on the sequence analysis. I think that's the question right now we have. We pretty much completed most of the questions. If you have any other question, you can add. The question is, will you show gene sequencing live in any lecture in the course? The answer is no, because it takes time. So there's nothing that you can see directly, because the reaction is done and then you run it in a sequencer gel. And after that, there's a data that is generated, and the data is analyzed to produce the data. So the sequence, right? These are called the raw sequence, and then you have to do quality check to ensure that whatever data that you have gotten is accurate sequence. So it takes time. So there's nothing that you can show live, but we'll be discussing the various methods in the lectures, but not necessarily a sequence of gel, a machine, or when it is, live demo, that we don't do. Yeah. Where to post doubt? I don't know whether you are referring to this live lecture or in the course. If it's a live lecture, you have the live discussion you already posted. If it is about the lectures, and there is a discussion forum in the course portal itself. When you log in, you'll be able to see that. You can add your questions there. You will get answers. I guess we are done with all the questions. If there are no other questions, we may conclude now. We will be having one more live sessions at the end of fourth week. We can again discuss. If you have missed any of your query, you can again join and we can discuss. One last question is whether you can work for a dissertation under your guidance, but this is not the forum to discuss that. That you can write to me separately over email, but this is meant for the topics that we have covered in functional genomics course. So I will not answer to this. You may write to me directly. In epigenetics, it is mentioned that behavior and environment change the way the genes work. Does that affect the information provided in the gene database? No, epigenetics normally defined as the changes that happens in the genome that does not affect the sequence per se. So, there are no changes in the DNA sequence, but the chromatin is modified. For example, DNA may get methylated, or the histone proteins that may be modified in different way. Therefore, this segment of the gene may differently function, and that is what we call as epigenetics. That will not show any difference in the genomic DNA sequence. Therefore, you don't expect any change in the database. That's why, the genome database, if there is a reference genome and that is put, you don't really make multiple sequencing done or anything. So it is mostly people look at the variations and mutations and so on, but human genome sequencing is not done again and again because that doesn't really make any new information. Right. So the other question is, can you suggest any articles, papers for learning more about genomics? So we will add some links that are available in the course portal. We'll add it, so you'll be able to read. Yeah, we'll do that. Okay, then I think we are done with this week's live session. I'm sure all of you are liking the course. So if you have any other query, then you can certainly post them in the course portal. We will take it up. Yeah. Thank you very much.